My name is Huaiyu. I'm an assistant specialist in Department of Anatomy and Neurobiology in the UC Irvine. And today I will explain you the procedure how to do the whole cell recording on the Drosophila brain. The key procedure of this preparation is how to decapitate the head, how to remove the brain, and how to remove the trachea system and the glia tissue from the surface of the brain. Today I will introduce you how to make a dissection solution for the whole cell recording in adult Drosophila brain. The first step is to make the dissection solution. Is really important. The key to make a good preparation for the adult brain. First, I will put 5 ml of recording solution in this centrifuge tube, and then here I have another small Eppendorf tube. I will add 1 ml of recording solution. In this tube, there have some cysteine, and then I will make this cysteine solution. I will pipette up and down to make a good cysteine solution, and then I will take 100 unit of pipette from this bottle, and then I will put this pipette into this centrifuge tube. And then you can see here the solution is cloudy. And then I will put another 100 microliter of cysteine, and then I just mix that thoroughly. 10 minutes later, the solution will be